So yeah, thank you so much for the nice introduction. My name is Danilo Bzdok. I'm assistant professor at the RWTH University in Aachen, Germany, and I like working at the intersection of neuroscience, psychiatry, and machine learning statistics. So in the following, what we are going to do is to look at what are the most important organizational principles that are typically assumed in the neuroscience community, and how can we actually go about and model them. This will take the first 2/3 of what we're going to talk about, and in the end we will try to how can we actually merge them to approaches and ways to think about brain organization that are usually separate. So the two core ways to think about how may brain function be organized in the brain are functional specialization, here on the left, and functional integration, which I try to depict in this picture. So what does functional specialization mean? It's this Brodmannian vision that there is a set of cytoarchitectonically specific brain areas that also have very specific receptor architectures that are specific to a certain patch in the brain. And it is this specific local infrastructure that allows to observe a specific class of computations, solve specific problems that the neighboring area might not be able to perform. But the emphasis is really on the mosaic segregation of the cortex. In functional integration, we take a completely different perspective and we say what is really important for brain function to emerge is the complex integration of all these functionally specialized regions. And different types of cognitive processes are really implemented by recruiting various types of these specialized regions, and it is this what is really important. So as you can imagine, those two are of course extremes of the same dimension, and the truth is probably somewhere in the middle, that neural processes are realized of course by a local infrastructure, but those have input and output connections to other specialized regions, and it is this interplay of bottom-up and top-down processes that makes brain function emerge. So the classic way to study brain areas and connectivity is actually through axonal tracing studies in monkeys. So this works the following way. You have a tracer dye that you inject typically in macaque monkeys, but also other animals, and they are taken up by the local membrane receptors of neurons and automatically transported by the normal axon protein machinery to various projection targets of that neuron. So the very important point is that this travels only one neuron typically. So what you do is you then sacrifice the animal, cut the brain into pieces, and look where did the color actually end up. And the beauty of this approach is that if you inject a color in, for example, the amygdala, and you observe color, for example, in the prefrontal cortex, you can infer that this entire monkey species, of which you perhaps just looked at one single member, is very likely to have a monosynaptic axonal connection between these two areas. So you can make very strong neurobiological inferences. So and here you see an example of what this looks like if you do it comprehensively here in the case of the cat. So you have the various brain areas here on the two axes. You can do this axonal tracing in the two directions, efferent and efferent. And you see one very important aspect of these things, and this is that every single area that we believe has a very unique infrastructure, cytoarchitecture, and so on, is also characterized by a unique set of input and output connections. Okay, so and this leads us to realize, so a cortical area, a specialized cortical area, not only has this very specific local infrastructure, but also the way it is set up to interact with the rest of the brain is also most likely unique to each part of the brain, at least in monkeys, cats, rats, and probably other animals. And here you see an example of this. So the question is, how can we go about and use that? So if connectional patterns, so the connectivity targets to the rest of the brain, change as you go from one specific cortical area to another one, then why do we not turn it on its head and we look for where might borders be situated by looking for changes in connectivity in the whole brain? And this is exactly what a technique does that is called connectivity-based parcellation. So the goal of connectivity-based parcellation is to come up with an atlas of the brain based on where do the connectivity patterns change. But the thing is, there are many different modalities in brain imaging that allow you to do functional connectivity analyses. I will just briefly introduce you to three of them. So this is diffusion MRI, resting state functional connectivity, and meta-analytic connectivity modeling. Diffusion MRI, this is how this automatic brain parcellation in brain imaging literature started in 2003. So this is the most frequently used type of connectivity in brain imaging for brain parcellation. And what it does is you specify a part of the brain, and then it tells you what is the likelihood in white matter to have fiber bundles that go from your seed region to other parts of the brain. So with various so-called tractography algorithms. So what you do is you go to your target area, and it tells you where and to what extent am I likely to observe tracts of axons, not single axons, but tracts of axons to all the various other parts of the brain. So diffusion MRI has a number of caveats, as all of them. Some of the most important caveats of MRI, diffusion MRI tractography, are that if you only talk about fiber bundles, it's difficult to say something about very specific axons. So in the axonal tracing in monkeys, we're really sure that there's at least one axon that goes from A to B. If you measure diffusion MRI, it's really difficult to say that because you can only say something about the mass of the fibers. It is also difficult to disentangle fibers that come very close to each other and then separate. And when white matter fibers kind of go away from the cortical sheath, this is typically something where we have a lot of uncertainty in diffusion MRI, and in the same way when they reach a connectivity target. So those are important caveats to keep in mind when you do diffusion MRI to come up with new brain atlases. Resting state functional connectivity is completely different. Both of these are actually instances of so-called functional connectivity or functional connectivity analyses. And that is in opposition to diffusion MRI because here we talk about white matter. It is structural, not axonal, but structural. And these are mostly focused on the gray matter. Both these types of functional connectivity have the tendency to measure not direct connections, so not monosynaptical. We cannot really decide that with these types of functional connectivity, but we believe that they typically capture polysynaptic types of connections. The most important difference between these two is that resting state functional connectivity is a measure of coupling between brain regions in the absence of a task. So what do I mean by task? Whenever I say task, I mean a psychologically defined experiment that hopes to recruit certain target cognitive processes and not recruit so much cognitive processes that the experimenter is not interested in. So resting state functional connectivity just measures the coupling in the brain in general. And it doesn't matter, or it is not looked at or considered closely what the subject is actually doing. In meta-analytic connectivity modeling, you also measure the functional coupling between brain regions, but during task states. And what is sometimes tricky to imagine between any task state. Okay, so it's different from so-called effective connectivity because you just measure functional coupling between a range of tasks. So this is the exact difference. This is task dependent, task independent, the functional connectivity that you get here, and the functional coupling here happens in various types of task states, so it is intrinsically task dependent. So one of the main caveats of resting state functional connectivity is now that as you just look at what's going on in the brain and you have less control of the mental space of the participants, it is difficult to disentangle those parts of the coupling that really contribute to cognitive processes and the proportions that may reflect noise or physiological factors. So this makes interpretation resting state functional connectivity tricky. In meta-analytic connectivity modeling, we have different types of caveats that are more technical. Typically, we only incorporate coordinates and not so much the complex 3D shapes of the neuroactivity that was initially observed. But it was really important to keep in mind there are not only these three, but even many more types of functional connectivity that allow you to do brain atlasing in a broad sense. But the conclusions that you draw in the end about what brain regions exist, where can I find boundaries between them, they intrinsically depend on the properties of the functional connectivity modality or structural connectivity modality that you use. And here are some examples. So there is a whole range of papers that have done various parcellations of various parts of the brain. You can do them whole brain. Yeah, you can just throw in the connectivity measures from voxels or parts of the entire brain, and it will give you a whole brain parcellation. But I would say most publications, they look at specific parts of the brain, such as the amygdala or only the visual cortex, and they operate by regions of interest. Some general things that come up is the closer we are to the early sensory areas, such as here in the visual cortex, the clearer usually the mapping from what we observe in brain imaging visual cortex parcellations and what we see in the animal literature. So the mapping is much more clear. And also in cytoarchitecture and animal research, there's a tendency that the differences between cytoarchitectonic areas or cortical areas, they're much clearer the closer you are to sensory input processing. So that means, on the other hand, the more you go up the neural processing hierarchy, so when you are in the inferior frontal gyrus here, so the frontal lobe, and we know that even if you look through the microscope, the boundaries between the areas are actually not that clear anymore. So it becomes even more difficult to really interpret brain regions that you come up with by performing methods for brain atlasing and brain imaging. So in the following, I will walk you through the process of how this actually works. So how do you come up with a parcellation of your favorite region of interest? So you start with so here we have a toy example. You start with a region of interest that is in brown here. And you need to imagine there's a number of voxels here. There are just 3 of them that we painted in yellow. But for each of these little locations, you compute a whole brain connectivity map. So you have as many connectivity maps as you have voxels in your region of interest. So this is what it looks like when you just compute one of these maps and you just ask, so we do it here in the example of meta-analytic connectivity modeling, but it would be in principle analogous to any other type of connectivity measure. In the case of meta-analytic connectivity modeling, you come up with a whole brain map of the co-activation occurrences. So if we look at one of these voxels here, how likely is each other part of the brain to co-activate across tasks? This is the question we ask. So if you just merge these, we come up with a smoother map here in the middle row, and then we perform a statistical inference, and we come up with the regions that are really statistically significantly co-activated with the specific voxel that we currently look at. So here you have 3 different whole brain maps computed by task-dependent meta-analytic connectivity modeling. And the point is that although these locations are rather close to each other in the brain, they lead to completely or to 3 maps that have a series of different connectivity targets. But we compute this for every voxel of the brain. So once you have this information, it is this coactivation matrix that you compute. So say we have 100 voxels on our region of interest. So in this case, in the motor cortex, then you would have 100 rows here, 1 row for each voxel, and then you basically flatten or vectorize the whole gray matter, and it ends up being one specific row in this cross-connectivity matrix. So it is this what you use as a basis for a series of so-called clustering algorithms. One of the most frequently used clustering algorithms is Kmeans clustering. And I will just give you a broad intuition of how this actually works. So here you have a bunch of points. Each of these points is the information of, so here we would have 2 voxels and how they are connected to each other. You compute 3 clusters. Okay, so one important aspect in brain atlasing is how many cortical areas do you actually need to assume or model. So in Kmeans clustering, you need to pre-specify that. In this example, you just say, I think there are 3 clusters in my target region in the motor cortex. So you randomly initialize the so-called centroids. And those are the centers of your clusters that you want to compute. In a second step, you then assign all the data points that are closest to that centroid, and they all become part of, say, cluster 1. And the same for the other centroids. Then you go back to the initial step, and you readjust the position of these centroids according to this cloud of voxels that have been assigned. So you need to imagine when we have 100 voxels in our target region, then we would assign each of these 100 voxels to one of the 3 clusters. And this is what you see here in these diagrams. And you iterate through these 2 steps of readjusting the centroids and assigning the voxels to the new cluster positions until you reach a convergence criterion. You can do this for different numbers of clusters, as I said. So this is what it would look like if you pre-assume there are probably 2 clusters in the motor cortex in my regions of interest. This is what you get if you say, no, I think there are actually 3. So the green one gets subdivided, and there's a smaller part of the green cluster. And this is what it looks like if you do 4. And there's a priori no limits. So one of the big issues in clustering, apart from choosing a certain number of clusters, is you always get a solution no matter how many clusters you assume. And that's a fundamental property of these clustering algorithms. And the trick is how can you inject your neurobiological intuition in getting around this choice. So other ways to perform clustering, I would just briefly mention those, is hierarchical clustering, also very frequently used besides Kmeans clustering. The way it works is you can start with as many clusters as you have voxels in your region of interest, so 100 in our case. And then you iteratively look for the 2 closest voxels and you merge them into a new cluster. Okay, and closest here means which of these voxels in your region of interest are closest in terms of their whole brain connectivity profile. And you can do this until you only have 2 big clusters left, and you even merge those. Okay, so there are 2 ways in which you can do this. You can do it bottom-up, agglomerative type of hierarchical clustering in this direction, or exactly the other way around, in a top-down divisive type of hierarchical clustering. The difference between Kmeans and hierarchical clustering, for example, is that here you can be sure that a different number of clusters kind of contains the neighboring solution. Yeah? So some cluster that you already have gets divided into two other clusters. So the different solutions of how this motor cortex is divided, they're compatible with each other. In Kmeans, that's not the case, just to give you an important property. Another way, this is spectral reordering. It is a semi-quantitative way to find structure in these whole brain connectivity patterns. And here, for example, it neatly allows you to say, well, if I arrange my voxels such that they neighbor close together, if they have more similar whole brain connectivity profiles, then you see, okay, there's a pretty clear evidence for two clusters in our region of interest of the motor cortex. So there are many, many other types of connectivity modalities, many other types of clusterings, but Kmeans clustering, hierarchical clustering, and spectral reordering are most frequently used in the MRI-based imaging literature. But those are not the only choices. Typically, you also have the question of distance measures and various ways to really create these whole brain maps. So there are a lot of knobs that can be tweaked. But luckily, at least in the motor cortex and the sensory cortices, there's usually a lot of agreement between what you get when you tweak one of these knobs a little bit. Okay, so once you have created the assignments of each voxel to one of the clusters, you can map those back into your region of interest, okay? So, and here in our toy example, with our region of interest, it indeed mapped back one cluster into what used to be this separate cluster here, and into this more posterior blob in green here. So it indeed separated the voxels into what used to be topographically distinct in the brain. But you need to imagine that this brain atlasing procedure itself, it has no notion of where each of these voxels actually belongs. Okay, so for the algorithm, it doesn't make a difference whether the voxels are immediately neighboring or in opposite parts of the brain. And if you perform types of functional profiling using databases, then they indeed have statistically significant function associations to different sets of cognitive processes. So this anterior one is more related to language, for example, and memory, whereas this posterior green one is much more related to action execution and other visual motor aspects. So it seems like, and this is also confirmed if you look at these locations with experimental fMRI data, it really seems like when you segregate this region of interest based on whole brain connectivity patterns, you come up with cohesive, they're not chaotic, they're neighboring sets of voxels that really do seem to represent functionally distinct compartments in the brain, which we can post hoc confirm by using databases to come up with functional associations and relating it to experimental fMRI data, and also seeing that working memory and tapping tasks differently engage these two parts of the brain. Okay, so this was more on the methodology. What's the general approach? Now we I skipped this one. Now we will talk about a concrete example, more hypothesis driven. Why should I care as a neuroscientist? Why could this be interesting to me? And we'll do this in the example of the so-called temporal parietal junction. So this is a pretty tricky part of the brain from various perspectives. So one of them is we don't really know where it is located. So we call it temporal parietal junction, but the word is more used in a functional. It's much more a functional notion than a topographical notion, which you can already tell from temporal parietal. Yeah, I mean that's a big part of the brain, and this is why people in different literatures refer to this potentially identical region, but with completely different terminologies. Yeah, some call it supramarginal gyrus, others call it angular gyrus, and there are many other ways in which posterior, superior temporal sulcus. There are many ways in which people actually refer to this region. And even worse, in separate literatures, such as sustained attention, sensory motor control, and the theory of mind literatures, people also assign rather different functions to what they think this temporal parietal junction that is already difficult to locate might actually contribute to. Okay, so we don't know where it is, and we have no idea what it does, but various literatures claim it's specific to something they are studying. So how can you go about and attack that using a brain atlasing approach? So what we did here is we computed the statistically significant topographical convergence in that part of the brain in 3 separate literatures. Okay, so this is the consensus location of where researchers studying sustained attention believe the TPJ is. And we have the same for the other 2 literatures, but the sets of papers are independent. So then you merge these into an overall target region. And then the question is, of course, if all these researchers are talking about the same phenomenon in the brain, then it should be pretty difficult to divide the brain region, this region of interest. Okay, we did it here by 2 different types of functional connectivity. We did it by meta-analytic connectivity modeling, connectivity-based parcellation, which is the task-dependent type of functional connectivity about measuring co-activation. And here we look at its brother, that is task-independent, resting state functional connectivity, connectivity-based parcellation. What you see here is, in the spectral reordering plots, it is less clear than in the motor cortex example we showed before, but there seems to be evidence for 2 different compartments based on whole brain connectivity data that indicate that there might be boundaries between cortical areas. If we map those back to the brain, this is what we get. So in 2 independent analyses, we find that there's anterior purple cluster in the right TPJ. And it looks pretty similar to the anterior purple cluster based on a completely different functional connectivity modality. So actually, if you look at it more closely, they agree in 90% of the time. So we can merge these into consensus clusters, and we use this to perform further connectivity analyses and functional profilings to find out what might these 2 regions actually be doing. So in a very conservative analysis, we find the following intriguing thing. If you ask, this may be obscure, but it asks a very simple question, what parts of the brain are connected to this anterior purple cluster during tasks, but also reflect negative coupling with the posterior TPJ at rest? Okay, so you're asking, what part of the brain is positively coupled with this anterior compartment that we discovered in the TPJ, treated differently in function and anatomy? And you come up with a set of regions in the mid-cingulate cortex anterior insula that we know to be frequently called the saliency network. On the other hand side, if you ask the analogous question, so what part of the brain is actually positively coupled with the posterior subregion in the TPJ, but negatively coupled with the anterior one, then you come up with the bilateral inferior parietal lobe and the posterior cingulate cortex, important nodes of we know to be the default mode network. So what is interesting about this is that we know that this network, no matter what people do in a brain imaging scanner, are most likely to increase in activity, okay? So whether it's an imaging viewing task or a working memory NBAC task, we are pretty sure by now that this set of regions are likely to increase the neural activity. And the opposite is true for this set of regions, because they have a very high tendency to decrease in neural activity pretty much no matter what the task is. So the point is, up to here, we automatically parcellated this controversial part of the brain into two subregions. And if you perform these rather conservative connectivity analyses with two different types of functional connectivity, we come up with tight couplings of large-scale networks that we know to be exactly opposite in their neurophysiology. We also performed the functional associations. Let me go into a little more detail this time. It basically comes in two flavors. The first is forward inference. So the way forward inference works is you assume knowledge of a psychological process or a cognitive process. So you say, I believe this process is true and it exists and it neatly maps on the brain. And then you ask, what parts of the brain are actually involved in that cognitive process? In reverse inference, you do the exact opposite. This is what we typically intuitively do when we look at neural activity maps. We look at brain activity, we have knowledge of signals in the brain, and then we think about what types of cognitive processes or psychological tasks might explain this pattern of neural activity. So there are complementary ways to think about how is the anterior purple and the posterior orange subregion in the TPJ actually related to function. And what you come up with across these two different ways to do functional association analysis is that this anterior purple one is actually much more closely related to types of psychological tasks and cognitive processes that are closely related to sensory input processing. Okay, they are much more related to processing something in the external world. The posterior orange one is much more related to kind of an environment detached scenarios. So where that may be influenced by your sensory environment, but that are mostly driven by information that you generate internally. So for example, when you imagine objects or scenes, or when you think about other people's minds. So again, this difference in neurophysiology of the large-scale network couplings of these two discovered candidate subregions, this is actually reflected in their functional associations that appear to separate according to externally or internally directed attention. Okay, so getting back to one of the trickiest questions when you do brain atlasing, and this is how many clusters to choose. And trust me, this is the preferred question by paper reviewers. If you perform some brain atlasing analysis, sooner or later, somebody will ask you that. Yes, okay, you have two clusters, but how do you know it's two? Perhaps it's three. And I like to emphasize that it's really important to appreciate that this is a very ill-defined question from at least two perspectives. So the first is from a more statistical or mathematical type of perspective. This so-called unsupervised structure discovery, such as clustering algorithms. Okay, this is true for the whole class of clustering algorithms. Doesn't matter which one. There is no perfect solution to the cluster number problem. Okay? Why? Because there are always various different ways to find relevant structure in data, especially if you have high-dimensional data, such as we in brain imaging. So this has been heavily investigated in the basic statistical literature, and there's no unique solution that has been found so far. From a more epistemological perspective, even if you had a statistical or mathematical mean to really decide on the one true best number of clusters, it still depends on the type of brain measures that you actually recorded. Okay, so perhaps the way you look at the brain, the glasses through which you look at a whole brain function, perhaps it neglects certain aspects that are important to the true number of cortical areas in the brain that you're trying to look at. Okay, so those are two fundamental boundaries that should make clear that it is pretty unlikely that there's this one true number of clusters if we deal with clustering algorithms and brain imaging. So, but just very briefly, there are three ways in which you can separate these heuristic types of analyses that can give you insight in what number of clusters might be more reasonable in my case. So you have aspects of topology. So this is what I mentioned, for example, about the difference between Kmeans, that between different choices of cluster number, the separations of a region of interest do not need to be consistent, but in hierarchical clustering, they are consistent. So these types of things. How consistent is my segregation across different solutions of cluster numbers? So there's this flavor. You have information theoretic approaches that are a lot based on entropy, so measures of chaos and measures of how likely am I to observe these data if I assume these cluster assignments, which is mutual information and so on. And the last one is perhaps the most intuitive, a lot of metrics to get an insight on how many clusters. They ask, to what extent are the voxels that belong to one of these 4 clusters more similar than if I compare any 2 voxels from different clusters in my current assignment, okay? So these are just some basic notions what the heuristics that are far from being something close to a statistical inference, but these ways to kind of approach the question, how many clusters might there be? And those are the common approaches. So for now, we've talked about how can you use clustering algorithms to perform brain atlasing that is targeted at brain regions. So everything we talked about up to now actually obeys the functional segregation vision of brain architecture, right? We talked about how can you find patches in the brain that have unique sets of input and output connections. So, but now you might ask, but wait, what about the functional integration where you put all the emphasis on the complex ways in which all these areas communicate? And this is what we will talk about next. So here, in this classic paper, it was not the first one that mapped sets of brain networks, but is definitely one of the most cited. And what I like to emphasize is whether or not you emphasize notions of distinct brain regions that are separate from each other, which we have talked about in the first third of this presentation, and the more functional integration perspective of brain architecture is actually a question of the algorithms that you use to find relevant structure, okay? So functional segregation is very closely related to these clustering algorithms, whereas if you come up with sets of large-scale networks that you derive from data, you don't use clustering algorithms, but types of matrix decompositions. So the ones that you probably have heard is independent component analysis. This is a very frequently used one, but there are many others that have the same basic idea. And those would be factor analysis, for example, principal component analysis, more modern sparse principal component analysis, or another modern way to frame these would be dictionary learning. But the point is, this other class of ways to find relevant structure in an unsupervised manner in brain data, they are much closer to notions for us neuroscientists related to functional integration, okay? So one of the properties that are different and that allow to kind of have this dichotomic way of doing brain atlasing is that here, each voxel in the brain is assigned not to one kind of phenomenon that used to be a cluster, but now we'll talk about components, but each voxel is assigned to a set of components. So if you look at these 10 networks, for example, in this component, you have a lot of IPS, intraparietal sulcus, on the right, but in that component, you also have some importance in that part of the brain, okay? So the assignment is not, there are no unique assignment, or you don't have a flat assignment. It's very important to realize that, okay? So each voxel of the brain is assigned to all components. And this is really important if you deal with functional integration, because all these different voxels in the brain, they probably relate in various ways by being connected to each other. Okay, I hope this makes sense. So, and one of the most important observations that came out from this paper is, if you perform standard independent component analysis, ICA, to map out what are the spatiotemporal components in the brain data that are coherent and that together explain the variation in my data, you come up with a set of these that look like this if you apply it on resting state functional connectivity, so the task independent type of functional connectivity. But what was really surprising in this one is if you apply it to task dependent data, those components are actually pretty similar. Okay? So the notions of macroscopical brain networks that you derive with ICA based on task independent data or brain recordings during defined tasks, they come up with large-scale networks that are pretty similar. And that raised the question of the so-called task-rest correspondence. Yeah? So if you talk about brain networks, you're suddenly much closer to, okay, how does this relate to the realization of complicated cognitive processes? And we found that the basic phenomena in the brain that explain the spatiotemporal variation, they are probably very similar when people don't do anything specific and are mind-wandering, and when people do something very specific. So the question is, how is it possible that these same things exist in the brain, and how do they mediate function? And that's something that you cannot investigate with clustering algorithms, okay? All right, so one way to look at this is the following. You extract a number of these large-scale networks. So here we didn't use ICA, but something very similar, sparse principal component analysis, but still the same philosophy. Each voxel gets assigned to one of 40 large-scale networks, and they are derived from resting state activity, okay? So we come up with a dictionary of large-scale networks that reflects what components in the brain explain the whole brain connectivity patterns, but in the absence of task. But then you take these to go to a battery of different tasks. So we did this with the data from the Human Connectome project and another big task battery called Archie from Neuroscience Paris. And what we observe is the overall pattern of, for example, gender judgments or trustworthiness judgments, which are two tasks that are rather similar. They do recruit similar components of these sparse PCA decompositions, such as one representing the limbic system, which makes sense, we are processing emotional information, a visual cortex, well, we have visual stimuli, and a sensory motor system. So all these three components are recruited during these tasks. But what is different between these two is actually to what extent is the default mode network, one of these things that consistently come up if we apply ICA to resting state data. The difference really seems to be in the default mode network, because this is a lot recruited in more complex trustworthiness judgments, but not so much in the easier control condition gender judgments. Okay, so this gave first hints how the spatiotemporally coherent phenomena, the components that we observe in resting state data, might actually relate to tasks. It appears that they get recombined in dynamic ways to subserve specific tasks. Okay, so the same atoms are combined in flexible ways to subserve specific function. If you slightly change the approach, you still come to this conclusion. But what we did here is you still derive a set of 40 large-scale networks that have weights in each of the voxels and that are topographically overlapping. Yeah? This is something that we couldn't do with the clustering algorithms that we talked about before. But this time, we derived them from task data. Okay, so you need to imagine from 18 tasks, you take half of the data, apply a matrix decomposition such as ICA or sparse PCA. You come up with a set of a dictionary of 40 networks, and then you ask, how are these related to the tasks? And what you see here is what is the most important network, if you ask for one, related to each of 18 tasks? And then you can ask, okay, but which ones are the most, explain the biggest variance in my data if I have 5 networks, and so on. So the thing is that if you derive a network dictionary from task data and not rest data, you also come up with an atomic set of the most important macroscopical networks in the brain. And you can also show that they relate in and are recruited in flexible ways to subserve specific functions. We have a very similar question here to the cluster number problem in brain atlasing into to come up with the regions, and that's how many components to choose, right? So, okay, we can come up with large-scale networks and derive these from resting state data or task data, but how do I know how many I need? And the answer is pretty much the same as in the clustering algorithms. There is no principled solution to this. There's just heuristic approaches that allow you to get insight into how many might be most appropriate for your data. But there's no solution that gives you an answer in general. So what we did here is, in 2 data sets with 18 different psychological tasks and 2 different ways to perform matrix decompositions, we asked, if I only have the information about 40, 20, or 1 of these networks of the recruitment, to what extent can I predict which task is present? Okay, so you need to imagine you reduce the data from the whole brain to 40 values up to 1 value. And then you ask, if I only know that 40 up to 1 network is recruited to this or that extent, how well can I predict which of 18 tasks is present? And the surprising thing is that across these 4 analyses, if you have knowledge of 5 macroscopical brain networks, then you can still, in 80%, which is just 10% less than the maximum, in 80% of the time, you can still say it's this one of 18 tasks. Okay, so this relates to the question, what are the degrees of freedom of the brain? If you want to put it from a little philosophical perspective, how many components that have consistent spatiotemporal structure do you actually need to reliably identify what mental operations a certain subject is doing? So the surprising thing is, if you just have knowledge about 5 values in the brain, you can have a pretty high certainty of which one of these 18 tasks is present. So the question that arises from this is, okay, if I can have knowledge of only the recruitment of 5 spatiotemporal components in the brain, is that better or worse than what I would get if I apply clustering algorithms instead and measure my brain activity in terms of clusters? Okay, so the question is, if you're dealing with fMRI data, and for whatever the reason is, and that might come up more often in the future as we have always more data, you only have a few values that you can use to describe your brain activity from tasks. What is more effective? Should I compress my information in brain networks that are overlapping and that are more related to functional integration? Or should I do some form of clustering and compress my neuroactivity information in these distinct regions that do not overlap with each other? So the point is, what you see here is, once you go back from this compression, okay, so you take the actual data from these 18 tasks in 2 data sets, and you compress it to 40 values. But the difference is, once you do it based on clustering algorithms and non-overlapping regions in the brain, clusters, and once you do it by mutually overlapping network components. And the question is, when you go back from these 40 values to recover, what did the initial task, so the ground truth whole brain neuroactivity actually look like? Which one looks better? So which one does actually capture the neuroactivity that I'm interested in? And the surprising result was, there's a pretty big difference in how well you recover between clusters and components. So if you segregate the brain into non-overlapping compartments, then the structure you capture in the data, it might still be able to tell the tasks apart, but it does not seem to capture the neurobiologically meaningful structure that actually describes the task, okay? And this is something you can see. If you back project from your compressed space with the only 40 values, to the whole brain map, and then you see, and you can also quantitatively measure this, that overlapping compartments seem to be a better representation of the overall whole brain connectivity activity patterns. Okay, so this is kind of a summary slide of this. Here we quantified this possibility to describe the complete, or hopefully very complete, whole brain activity by clusters or components. And for 18 tasks in 2 data sets, for 4 different matrix decompositions, so networks, and 2 different clustering algorithms, so clusters and regions, and we measure how well can I actually recover the initial whole brain information. And the thing is that whenever you use overlapping compartments in the brain, you're much more effective in capturing structure that is likely to be neurobiologically meaningful. Okay, so and up to this point we looked at this kind of dichotomic view. Okay, so I can either look at brain regions, look clustering algorithms, and discover disjoint compartments in the brain, or I can, which seems to be better in fMRI data, which might not be a judgment about the brain biology itself, but if I apply matrix decompositions, such as IC, sparse PCA, PCA, and so on, and I discover compartments in the brain that are mutually overlapping, it seems to be a more effective representation of our fMRI signal. But the question now might be, how can I actually combine these two? So what you should be kind of skeptical about is, does it actually make sense to say, I want only discover regions that are totally separate, or I specifically emphasize that these components are overlapping and so on. And what we know to be most likely the case is what became known as the multi-to-multi mapping. What do I mean by multi-to-multi mapping? Each part of the brain, so let's say each cortical area, is recruited not during one, but a whole set of different types of psychological tasks. On the other hand side, every specific psychological task also recruits a specific set of brain regions, right? So if you go to one specific region, it's pretty hard to tell, okay, what is this one thing that this region does most? Yeah, this is probably an overly simplistic question. So what is more likely to be the case, and that is emerging and very well summarized in this paper, is the view that there's something like cognitive network components. Okay, so there might be recruitments of cluster and network aspects that are flexible and that are specific for certain tasks. Okay, so a given brain region might be recombined in various ways to subserve certain psychological processes. So the question is now, how can I capture this higher order interaction structure without being constrained to only looking at functional integration or only looking at functional specialization? Because what is very likely to be the case is that a specific cognitive process is probably somewhere between the extremes of a functionally specialized region and the high importance of coupling of these regions. So I'll just give some examples of how you could actually do this. So one of the ways is you do not use clustering are the usual ways of matrix decomposition, but something that is called the autoencoder. So the autoencoder is not so much used in brain imaging so far, but we know from formal mathematical proofs that it can discover structure that is related to clustering algorithms in brain regions, but it can also discover structure that has much more the properties of matrix decompositions in brain networks. Okay, so you probably see what I'm getting at. If you use autoencoders to find relevant structure in brain activity data, then you are likely to find components, let's say cognitive components in the brain, that have aspects of regions and aspects of networks. So you want to be in between the classic organizational principles. And the way this works here is you integrate this into actually an inferential problems. So it's a supervised algorithm. You just discover structure as one part of this model. But what you actually do is you want to find out how do I need to assume the relevant compartments in the brain? What form should they have if I want to be really good at distinguishing, for example, the 18 tasks we were talking about, okay? So you're not asking what's the relevant structure in the brain in general. You're asking how can I be exactly in between the two classic organizational principles with the aim of telling these tasks apart. And this is how you can discover structure and infer relevant functional compartments in the brain at the same time. So just one example what this looks like. So here we took experimental data from an empathy tasks. So just imagine there are 5 different conditions and they all differ in stimulus and task, but they're all related to empathy. And what you ask is, what are the 10 cognitive components that together best predict and allow me to reliably predict in new brain images which one of the 5 empathy conditions are actually present, okay? And the interesting observation is you come up with things that look much more like local brain regions, such as the one here. So there are high weights in the medial prefrontal cortex. There are some other locations, but there's a focus in this medial prefrontal part of the brain. But you also find components that look more like a brain network, such as the component 1 here in the visual cortex or the bilateral auditory cortex. So the point is that we do not need to constrain ourselves to disjoint regions or only look at networks, but we can find structure, so 10 cognitive components that are specific to predicting empathy and not various tasks. Yeah, we can discover the cognitive components that are specific to difference in empathy conditions, and they are in between organizational principles. Another flavor to be in between chairs of regions and networks is what has been published a couple of weeks ago, and that is based on notions of the geodesic distance. So it starts out with the observation that if you look at these target regions, you measure how far are they away from each other. You discover they're equidistant from the motor cortex, but also early sensory cortices, such as the visual cortex and the auditory cortex. And funnily enough, the set of regions is what we know to be the default mode network. Okay, so what these investigators discovered is if you look at a different notion of how connectivity and regions relate to each other, then you can explain the default mode network as the potential top of a neural processing hierarchy that is equally distant from the early visual input processing, but also with the same kind of distance to motor output processing. And the way they do it is they come up with relevant structure in the brain. So again, we have types of cognitive components, if you will, but they are derived from what is the connectivity gradient in each of the parts of the brain. So they look at a different notion of how two locations in the brain are actually related to each other. And that allowed them to find gradients of changes in connectivity as an organizational principle that might be complementary to what we have been doing before. And they find that the large-scale networks that we know and love, such as saliency network, dorsal attention network, and so on, they really seem to be organized along, they neatly are situated along these connectivity gradient dimensions. So this is a possibility. And yet another way to not be constrained to distinct regions or intertwined networks and clustering or matrix decomposition is this approach here. So the authors set out to come up with an atlas that is cognitive. So it starts out by the following observation. We typically do reverse inference when you look at brain images, right? So we look at neural activity and we think, oh, that person is probably doing a pain task or something. So that is what has been long discussed to be dangerous, reverse inference. Russ Poldrick published a very influential paper on this roughly 10 years ago. And these authors tried to come up with a valid reverse inference atlas. Okay, so when you look at a part of the brain and you needed to assign that part of the brain to mostly one psychological function, which one would that be? Okay, so this typically doesn't work with the standard statistical approaches that we use, but they came up with a completely different machine learning approach to make it possible to derive maps that are actually allow for valid reverse inference, okay? So what you see in orange here, for example, those are the parts of the brain that across dozens of cognitive labels are together, most often really reflecting that a facial processing task is happening. And they found other whole brain patterns that are related to different sets of tasks. So the point is, you can also look at this from a more cognitive perspective and ask, can I design an automatic way to come up with atlases of functional compartments in the brain that are most specifically, not uniquely, but most specifically related to the cognitive notions that we typically use in experimental brain imaging. And this is very well possible. So as the last conceptual slide, we talked about functional specialization a lot and functional integration a lot, but this is most likely not the end of the story. There's something that is underneath these two things, and that is the time dimension. So for a long period in the history of neuroscience, we believed that electrophysiological oscillations, they are really chaotic, and we don't really know to what extent they really reflect the targeted processing of problems to increase reward and reduce pain in a certain organism. But in MEG literatures, it's really becoming clear that the different frequency bands are very conserved across mammalian species, and they really appear to reflect specific functions of neural processing. So in the future, the goal probably is not only to be flexible in whether we adhere to distinct regions or intertwined networks to explain how complex function comes about, but we also need to incorporate the complex time dynamics in which these functional compartments interact and how these are specific to certain brain functions. And those are the things that we investigate in SP2, in the Human Brain Project. and very specifically also the individual brain charting projects. And there are many exciting projects that are related to this conundrum. And I'm happy to take any questions.